Why the line is like that? Okay, I hope that the line is okay now. Don't know lah why the line is like that. Okay, maybe I give some time, you come in. Aiyo. Okay, I will start 8:25. You can share the list new link to your friends. I hope that letter is totally smooth. You Okay, so now I proceed to the third experiment. So I don't know, I stop at where, so I just repeat again lah. Okay, so actually this experiment, third experiment is why got this third experiment is that the scientists not believe that the second experiment is okay, it is true that the genetic material is carrying the no DNA carrying the genetic material. Many scientists does not believe that and does not mereka tak percaya lah keputusan itu. So at that time got another two scientists called Herschel and Chase. They finally prove this. And then after that, nobody else give objection about that the DNA carry genetic material lah. So all of us is believe that and we until now lah. Until now. Okay, so this experiment actually totally different from the previous experiment. The protocol and the procedures are totally different. So the microorganism that he used is bacteriophage. It used the one type of virus called bacteriophage already. So if you see that this virus is look like this, dia macam diamond shape kan. So it is one type of virus called bacteriophage. Why he use this? Why he use this? Normally it want to make a differences between the previous experiment lah. Because previous experiment got someone say that only bacteria can be used, so can be applied. So some others organism cannot be applied. So cannot. So these two scientists they use another uh microorganism lah. And then they found that also because our previous experiment already proved that right, it just want to double confirm only, already proved that DNA is the carry genetic material. So bacteriophage also carry DNA and also it can make an infection to the E. coli bacterial cell E. coli. So it already can make an infection to the E. coli, so that when use bacteriophage is more suitable lah. More suitable and more clearly to see the effect. Okay, so how this experiment is do is it is firstly the bacteriophage is cultured in the medium that contain radioactive sulfur and also radioactive phosphorus. So these two experiment is conducted separately. They are conducted separately. One of the medium is cultured in the one of the bacteriophage is cultured in the radioactive sulfur, contain radioactive sulfur, and another one is cultured in the radioactive phosphorus. So if you see that it will give the radioactive sulfur is pink color, and another one is blue color one. So why they want to do culture in these two different culture? So radioactive sulfur actually it will incorporated into the fudge protein only, meaning that the body, its body. So because bacteriophage body is made from protein, it is made from protein one. So normally it will put this sulfur only can incorporated to the protein only, its body only. So maksudnya DNA is not completed. So DNA we cannot see the color one. We only can see the color pink color is about its protein. Its protein give the pink color. And then this one phosphorus blue color is it will incorporated with the DNA. It will bind to the DNA one. So meaning that that protein is colorless. We cannot see why. We see blue color because of its DNA. Okay, that next E. coli then will emerge into the then E. coli will be emerge into this culture so that bacteriophage will start to infect the bacteria cells already. Dia akan start buat infection lah. It will start to infect. So how it's infect? They know that when bacteriophage infect the E. coli, it will release the DNA. And its body will not enter, will not infuse into the E. coli one. Only the DNA will enter the E. coli. Protein body will not enter the E. coli one. It will remain at the outside one. So you see here, only DNA, fudge DNA is infected, infecting the cell. The body is not infected. The body will remain at the outside. So if you see that later, then after infection, after many few hours, after the infection is done, then they will go through the agitation. So hormonization lah, blinding. Why they want to do a blinding? Because they need to separate the E. coli and also the bacteriophage. So after that, they centrifuge the mixture so that bacteria form a pellet at the bottom, and free fudge and fudge part which are lighter will remain suspended in the liquid. So if you see that, see that only the supernatant will be the fudge body at all, fudge part, and then the pellet will be the bacteria E. coli. Okay, so if you see this, when the bacteriophage is immersed in the radioactive sulfur, so pink color. So if you see that only the supernatant remain pink color, the pellet is still remain cocoa color, brown color lah, or remain colorless, white color. Normally is white color. So you see that only supernatant is purple color. So meaning that actually DNA will not maksudnya protein will not infect at all. Because we know that when bacteriophage need to infect DNA, infect E. coli, it will inject DNA directly. And then this DNA will change the sequence, right? We know that DNA will change the sequence of the E. coli lah. So we know that protein will not infect at all. Protein tak ada infect punya. Protein will remain at the outside one. Only the DNA will infect into the E. coli. So if you see here, you found that pellet is colorless, totally colorless, and then only not pellet, supernatant is colorless at all, and then the pellet will become blue color. It will become blue color one. So blue color meaning that DNA is already infected to the E. coli. So fudge DNA already enter E. coli already. So that is why the pellet will show blue color. So in all this experiment, what are the conclusion? You can know that DNA will make an infection into the cells, not the protein. Protein will not make an infection one. Only DNA can make an infection and change the sequence of the cell, change the genetic sequence of the cell. So these are the explanation you can make yourself. Okay, so the final conclusion that we can make is DNA will be the molecule that carry genetic information. So finally, from this experiment, we can conclude DNA carry genetic information already. So this is the three experiment made by the 6 scientists. They did finally prove that DNA carry genetic information. Okay, so that's all for experiment part. This is just the introduction to full proof that DNA carry genetic information only. We are not talk about DNA replication yet. No DNA replication yet. So hopefully guys can understand it. Okay, so if you have any question, you just type in the chat box. My mic got no sound. !aiya! Okay, so I already talk. Okay, I already finished the BGM. Not BGM. Already experiment. So hopefully you guys can understand it. Okay, so next I will proceed to the DNA replication. Okay. Okay. Not this. Okay, so next is about also DNA replication topic. So, okay. So for this one is to want to know the model of a DNA replication. So what are we already know that DNA carry genetic material. So how DNA replicate? Bagaimana DNA tu boleh buat replication. So that is what we know, what we need to know. So actually got one scientist called Watson and Crick. He did the DNA replication model. So I think this scientist you are familiar, right? Sem 1 you have heard it, right? So he was also the scientist create the DNA model one. DNA double helix polynucleotide chain DNA model, right? Inilah orang yang Watson and Crick he proposed that model, and also he also proposed the DNA replication model as well. So so if you see this diagram, you can see that it proposed three models: conservative model, semi-conservative model, and also dye-specific model. Okay, so what are these three differences model? Okay, so in example, for conservative model, it is the model like this. So kalau lah ini this is one DNA, ini salah satu DNA. For conservative meaning that after the replication, so after replication actually it will produce two DNA strain, right? After they doing a replication, kan replicate kan copy and paste. You can see that copy and paste. So it will produce two DNA strains. So for conservative, it shows that one of the daughter strain is totally same as the parental strain one. Totally same, 100% same. And another strain, I use different color, it is totally different. Ha, totally different one. Yang ni 100% same. Yang ni 0% same. Totally tak sama. So you will see here why you see that it is more light color. So meaning that this one is same as the parental DNA strand, and then this another total strand is totally different one. Totally different one. So this is the one of the model we call as conservative model. Maybe DNA replicate like this, maybe. Because he also don't know which model actually is the true DNA replication model. He also don't know. That time he also don't know. He just estimate only. What estimation, make a hypothesis only. But which one he also don't know. So this is the first model. Okay, another model is semi-conservative model. Okay, what are the semi-conservative model? Is it also have the one of the parental strand, and then it replicate how it replicate, meaning that got one strand polynucleotide strand is come from the parent one. Salah satu strain tu polynucleotide strain tu dari ibu dia, dari parental dia. And another strain is new strain, meaning that dia ada satu strain is come from parent and another strain is totally new strain, but it is complementary to each other lah. But it is totally new one. Not come from the parents one. So this is the second model, semi-conservative model. Why is called semi? Because it is half the power. So the two traits of the parental molecule separate and each function as a template for synthesis of a new complementary strain. So meaning that each strain is act as template. So if you see that for this one, the two parental strain is reassociate after acting as template for new strain. So meaning that when this conservative they imagine that when they do a template as the template, and then after that they combine together. So if you know that, so actually DNA replication is like this lah. Normally in general lah. Later I will explain it. In general dia macam ni. Dua DNA strand, right? Macam ni. Lepas tu mereka akan dissociate, akan potong, become single strain, and then they will act as the template, and then got another, and then it will produce another strain one at here. Akan produce a newly strain. For conservative, meaning that after it become template, they akan separate balik and then combine to the original one. But for semi-conservative, it will remain unchanged already lah. Tak berubah already lah. And then another model is dispersive model. Okay, so this model is more tricky sikit. Dia awak boleh nampak macam pelik sikit. So actually this model is about also got one DNA strand at here, and then this model is each strand of both daughter molecule contain the mixture of oldly and newly synthesis DNA. Meaning, campur je. Meaning, if you do this, dia akan jadi macam ni. Macam ni. Why like that? Because you need to another color. Meaning, dia ada sini, ke sini, ke sini. Lepas tu, dia ke sini, ke sana, ke sini. So, maksudnya dia rojak. Dia rojak punya. Memang rojak punya. Dekat data strain ni dia memang rojak punya. Ada parent dan juga new strain one. New nucleotide one. So totally is rojak. So this is why we call as dispersive model. So as a scientist, as a professional scientist, we cannot say that all these three models are about DNA replication. No, only one of the model only. But which model? Actually, Watson and Crick, he also don't know. Dia pun tak tahu lah. These two scientists just propose the model only. So, which model? He also don't know. So, who proved the model? So, which model appear? Who proved it? So, we have another experiment. Number 4. Fourth experiment. Experiment keempat. So, who proved it? These two scientists lah. Ni dua scientists ni. Mattel, Meselan and Frankenstein. So these two scientists, they did the experiment to prove it that the model. So finally, I just give the conclusion that semi-conservative model is the model for DNA replication. The conservative model and the dispersive model are totally rejected. So how to prove it? Macam mana prove it? Tak boleh cakap je. Nak prove, para saintis kami kena tunjuk bukti, bukan cakap syok je, kan? So as the profession, you need to prove it. So how to prove? They did this experiment. So if you see this experiment, it more macam awak tak tahu dia lukis apa. So if you see that this experiment is dia guna also E. coli. Normally we use E. coli lah. You also use the bacteria E. coli. So bacteria E. coli contain DNA, right? So the E. coli will emerge in the radioactive nitrogen. But carefully, there are two radioactive, which is light and heavy and light. This one 15N is heavy punya radioactive. Yang ni 14N is light punya radioactive. Both these, when the bacterial culture is immersed, this radioactive will incorporate with the DNA of the E. coli and will give the either heavy band or light band. So actually no color lah. Actually no color. We don't. This one it use color lah more clearly. But actually no pink color and blue color one. So in examination, please don't write pink color and blue color. No pink color and blue color one. This is just the one to demo for you. Want to make you see more clearly. Between these two difference. So this one pink color one is actually called heavy medium, and then this one is light medium. So firstly, E. coli will be cultured in the 15N heavy medium. Okay. After that, they do an analysis. They will found that only one band. You will found that only one band at here. So it is more at the bottom side. Why? Bottom side. It is the heavy band. Why heavy band? Because it is heavy. Heavy DNA. So the mass is more heavy. So it will at the bottom side. And then after that, after culture few hours, few days, normally is 24 hours. Okay, after 24 hours, it will culture again. This culture will be addition with the 14N light medium. Dia akan campur lah. Ini dia akan campur dengan 14M medium. And then also culture about 24 hours. Okay, so after 24 hours, they do the analysis. They found that the band obtained is at the middle lah. Dia bukan heavy lah. Dia at the middle one. Dia lebih tinggi sikit lah compared to here. So if you see here, you will found that it is look like this one. Intermediate density. Okay, for now, they need to prove. So after that, this media already contain heavy and light media. They culture again 24 hours. 24 hours. After that, it found that they do the analysis. It found that got two bands obtained. Ada dua band obtained. It is dua band obtained. It is one is light band, another one is heavy band. Another one is you can say that is intermediate band. Not heavy, bukan heavy. Not heavy, bukan heavy. Okay, it found here. Okay, so for now, we need to eliminate already. So if you still ingat for conservative, maksudnya anak DNA tu, dua-dua DNA akan one is totally same, another one is totally not same. Like one is like this, and then another one is this. Totally not same at all. So meaning that no hybrid at all. Tak ada hybrid. No hybrid at all. So if you see here, it already have hybrid band. intermediate. Meaning got intermediate meaning that the DNA got dia ada campur. Dia ada campur punya. Ada N15 and also N14 punya. So meaning that conservative model totally out. Kerana conservative model tak ada hybrid punya. It does not contain any hybrid. Kalau conservative model, dia akan hanya dua band. Either one band is light band and another band is heavy band. Only these two band already. It will not have the intermediate band one. So directly we eliminate conservative model. But dispersive model, we cannot eliminate it. Why? Because dispersive model, it will have the intermediate band one. There are the intermediate band one. So maybe this kind dispersive model can be obtained. Maybe it can be obtained. Okay, so after that, you already eliminate conservative. So I got two choice only, semi-conservative or dispersive. So after that, they culture 24 hours again. So make a E. coli, make a replication again. So they found that got two bands occur also. One band is light band, another band is heavy, another band is also intermediate band. So this means that if dispersive, however, whatever you do the DNA replication, it will only totally hybrid. Memang hybrid punya. Meaning it will only have one band of it only. Mean it is intermediate density band. It will only have one band kalau dispersive. Dia tak akan ada dua band punya. Tak ada punya. Kalau dispersive lah. But it show that it have two band. One is light band, another one is dispersive. Not dispersive, intermediate one. So meaning dispersive model totally out juga lah. Karena kalau dispersive, if dispersive, it only have one band already. One intermediate band only. Kalau dispersive lah. But sorry, no. Hybrid, no, it will have two band. So meaning that it is only semi-conservative model only. So finally, it proved that these two scientists proved that semi-conservative model will be the model that will and DNA undergo DNA replication. DNA replication will follow this model. Others two model they are not follow. Not logicable. In real world. So okay, so these are so actually this one is same. I take from Campbell lah. Ini saya buat ambil dari Campbell. Okay, so actually the result is same lah. So actually this one it will give you lah. So if notice that in first replication, if conservative model, it will have one light band, and one heavy band. So if you notice that the first replication it only have one band only, right? Kalau macam ni, dah ada satu band only. So meaning that this one is totally eliminate conservative model already lah. And then this one you see that it got two bands. One is less dense and another one is more dense. More dense. So for this one, we also can eliminate dispersive only. Dispersive because why? Because if only dispersive, meaning that it only have one band only. Hanya satu band je. Tak ada dua band punya. Kalau dia ada dua band, memang bukan dispersive. So, okay. So finally, we only have semi-conservative model. So in ini explanation, you can read yourself. So this one conclusion is the semi-conservative model will act as the DNA replication model. So other conservative and also the dispersive are totally rejected at all. Okay, so these are the 4 experiments to prove that DNA replication follows semi-conservative model. Okay, so that's all for this experiment. Okay, so next I will proceed to the mechanism of DNA replication. So these are the main hot topic lah. Many people will read, but not say that the experiment is not important. Not important. Okay. I hope my slide is clear. Everyone can see the slide. Okay. So this one is about the DNA replication mechanism. Okay. So actually, if you see here, this is just the overview of the DNA replication. So you see that this one is the original double-stranded DNA molecule, right? Original one. And then after that, it will become the single-stranded. The double-stranded will be split out, become single-stranded, and then it will become the bubble form. You see that it will become the bubble form. This is what we call as bubble, bubble form, replication bubble. Okay, after that, there will have this original form, original DNA, parental DNA will act as a template, and then another DNA strain will be formed at here. Dia akan form dekat sini. Form dekat sini. So after that, they will form until they form together. This bubble will become big and big, and then until it form here like this. Two daughter DNA molecule will form. So you see that only one strain is from the parents and another strain is daughter. So it is totally different one. It does not totally different from the parent one. So this is the main overview of the DNA replication, meaning got one double-stranded DNA just split into the single-stranded, and then the another daughter-stranded will be produced when we use the parental template act as the template, and then to form the two daughter DNA molecule. So this is just an overview. So we need to know that how it is split into the single-stranded and how the DNA nucleotide added into it, and then how to form the final product, two daughter DNA molecule. Okay, so firstly, first step is to from supercoiled DNA to circular DNA. So in our cells, our cell is arranged in supercoiled form like this one. Ha. Memang like this one. So this is more stretching form and more complex form. So if you want to use this form to undergo DNA replication is nonsense. Tak boleh. Memang tak boleh. Because yang ni it is more packed together. It is nonsense. Tak boleh, langsung tak boleh buat DNA replication like this. So we need to become a circular DNA. So what are the enzyme that possible to become like this? DNA topoisomerase, if I'm not mistaken. DNA topoisomerase. This one. DNA topoisomerase. This enzyme, it will break down the supercoiled structure of DNA, become the circular DNA. So how it break down, dia akan cut at here. It will break down at here. So it will become the circular DNA, which more can be to form the baru boleh buat DNA replication. Kalau macam ni memang tak boleh. Okay, so these are the first step. So next is second step. Second step is to unwind the double helix DNA structure. So if you see that this is still the circular DNA still is double stranded. They still remain double-stranded. So if we want to do a replication, we need to unwind the double-stranded DNA structure. So how to unwind it? We will have the enzyme called DNA helicase. So helicase, DNA helicase. DNA helicase. So this enzyme will unwind and separate the parental DNA strain. So this very clear function. Dia akan potong, dia akan separate dia lah. So kalau DNA dia memang macam ni kan, so dia akan jadi macam ni. Tapi dia tak macam directly macam ni lah. Normally the enzyme will separate like this one. Like this one. So the DNA helicase is at here. Dia akan potong macam ni. This is a DNA helicase. Dia akan potong the unwind the double stranded DNA structure become like this. So become the single stranded lah. Okay, From here we also have another enzyme, not enzyme called protein, single strand binding protein. So this protein is to stabilize the unwound parental strand. So normally if the DNA is already unwound, become single stranded, so it is not very stable lah. Right. Kan awak sudah berpisah macam awak dengan boyfriend girlfriend awak, sepatutnya sentiasa setiap hari together, tiap-tiap hari together punya, tiap-tiap hari pergi makan, pergi shopping, tengok movie kan. Tapi tiba-tiba ada itu DNA helicase ni potong awak, cakap kamu tak boleh dekat dengan awak. So we need to have a, so maksudnya awak rasa sangat sedih kan, marah, sedih. So want to stabilize perasaan awak, another protein called single stranded protein. It will stabilize the parental strand to become more stable lah, not to if the DNA strand is not stable, then it will finally become like that. So if the DNA is like this, normally it's already cannot make a DNA replication mechanism already lah. So because DNA replication mechanism is one by one, because you know that DNA strand is very long, it will not totally cut down the double stranded DNA strand become single stranded. No, it will make a replication one part by part. Macam ni, you see that it will make a replication here first. Dia akan buat sini, lepas tu perlahan ke sini. Lepas tu yang ni dia akan stabilize lah. So kalau di sini DNA replication already finished, dia akan mula-mula become the double stranded again. So this is the function of the single stranded binding protein. And then this one is topoisomerase. So topoisomerase is to break, sever, and rejoin the parental DNA ahead of the replication fork, relieving strain caused by unbreeding. Okay. So they are already got 3 enzymes already you need to know. Topoisomerase. So there are many enzymes. I just want to mention there are many enzymes. Topoisomerase, helicase, and also single-stranded protein. So there are already 3 enzymes in this reaction already. So please alert, you need to memorize all these enzymes and also the function. This is only the second step only. Okay, this explanation so you can read yourself. Okay, formation of leading strain. Okay, so in DNA replication mechanism, if you see this diagram, you see this diagram more simple, right, compared to the outside one. So if you see here, if the when the DNA helicase already do the what unwind the DNA structure, double stranded structure, it will form the bubbles like this. And then this bubble will contain origin of replication. So this part is we call as origin of replication. Meaning what we call as origin of replication? Meaning that the addition of DNA nucleotide will start at here. Dia akan start dekat sini. Bukan dekat sini bukan simply dia start je. Bukan at the DNA nucleotide not simply one at one. Tak boleh punya. Dia ada peraturan dia. So origin of replication meaning it will start at here, same at here. Okay. So how it start? We got one enzyme called primase. Primase. So we will have the enzyme called as primase. So this enzyme, ini enzyme, primase. So what primase do is, will synthesis the RNA primer first. Dia akan synthesis RNA primers first. Meaning ini is RNA primer. Siapa yang sintesis? Primase. RNA primase lah. RNA primase will synthesis the RNA primer first. So this primer is actually very short. It is very short only. So this one is RNA, not DNA not DNA, bukan DNA. Ini RNA. Because why you need to synthesis RNA primer? Because the DNA nucleotide cannot directly added to here one. Tak boleh directly added to here. It needs the other nucleotide first. Dia perlu ada satu nucleotide first. Baru DNA nucleotide baru boleh add here. So after RNA primer is formed, we will have another enzyme called DNA polymerase 3. Please, 3. DNA polymerase 3, not 1 and 2. You need to specify it. If you in examination, you write 1 or 2, or you just only write DNA polymerase, sorry, no marks at all. So this enzyme will bind at here. Yeah, it will bind at the RNA primer and then it will produce the leading strand. Dia akan menghasilkan leading strand at here from this direction. So leading strand will form at here. So how it's formed? It will add nucleotide lah. So DNA polymerase 3 ni dia bind at here and then the addition of DNA nucleotide akan tambah ke sini using this parental as template. So what mean that by template? Meaning kalau sini is A, maksudnya dia akan campur T lah. Complementary. This C dia akan campur G at here lah. So dia akan campur masuk ke sini. Okay. So this is what we call as leading strand. This is called as leading strand. So like here, you see here, dia akan bind at here. DNA polymerase 3 bind at the RNA primer, and then it will synthesize from toward the replication fork. So replication fork, what is it? So this one, these two part we call as replication fork. It will form the strand toward the replication fork. Bukan dari sini bukan tak boleh. Why tak boleh? Bukan. Di ini direction ada mechanism lain. So the direction of the strand is 5 to 3. 5 ke 3. Bukan 3 ke 5. You need to know 5 to 3 direction because if you see that this one is 5 pi to 3 pi, right? And then this one is 3 pi to 5 pi. So meaning that if you want to form the DNA, you know that DNA is arranged in anti-parallel side, right? So this already 5, 3. So meaning that this one will be start to 9, 3. This one will be at 5, and then this one is 3. So 5 to 3. Because you know that DNA is arranged in anti-parallel form. So the direction is 5 to 3 direction. So this is the formation of the leading strand. So same as here. So for here, so same as the opposite side. So opposite side here, it will form the RNA primer first, very short RNA primer, and then it will form the leading strand. So it will form it here. So until replication fork so this is the replication fork, and then stop. So this is the formation of leading strand. Leading strand. Okay, next we will proceed to lagging strand. Okay, so how lagging strand form? So this explanation. Okay, so if you see here lagging strand, so leading strand already formed. So you imagine that still remember that how about this direction? So this direction we will form lagging strand. Yang ni direction ni, yang ni empty ni, we will form lagging strand. So this one will form for against from the against from replication for what? Meaning that our primer, our RNA primer will start at here. Kami akan start dekat sini. So I use different color lah. So yellow, okay. It will start at here against the replication fork one. Bukan against replication fork, bukan dari sini ke sini punya, bukan dari sini, bukan. Dia akan dari sini ke sini. Okay, so if you see here, it will also form the RNA primer. So I already mentioned that DNA nucleotide cannot simply add the nucleotide at here without any nucleotide. Tak boleh punya. So it require RNA primer. So RNA primase will bind at here again. It will bind at here and then form RNA primer at here. Very short one lah. Very short. Okay. After that, the another enzyme we call as DNA polymerase 3. DNA polymerase 3, same enzyme, this one. It will it will bind to the RNA primer to form the strand at here. Form at the strand at here. Okay. And then after that, there will also got another primer also at here. And then another strand at here. Right here. And then you will see that. Okay, there are another primer and then another strand at here. So why does this happen? You will see that the major difference between leading strand and lagging strand is leading strand is formed continuously. DNA polymerase directly add nucleotide until toward the replication fork. But lagging strand, no, it will not form continuously. So why? Kenapa? Kenapa tak boleh form continuously? So this one is due to the maybe there are many origin of replication, because this one at here got one origin of replication, but actually at another part we also have another of origin of replication. So meaning that the lagging strand will form the DNA polymerase will add the nucleotide until it comes through the another primer. So it already it will detect got another primer already. So it will stop adding already because it already touch an RNA primer. This one is RNA primer, right? RNA primer dia tak boleh pilih punya. DNA polymerase hanya boleh tambah nucleotide je. Dia tak boleh tukar punya. Okay, so this one we call as Okazaki fragment. This one yellow punya we call as Okazaki fragment. Bukan lagging strand. We use the name Okazaki fragment. Okay, so you see here. Okazaki fragment. So all of these are Okazaki fragment. Okay, next. What are the next is we need to change the RNA primer become DNA nucleotide. Kan, ini RNA. Kami nak tukar kepada DNA. So how to change it? There are another enzyme. So another enzyme we call as DNA polymerase 1. First type. DNA polymerase 1, it will bind to the RNA primer and then replace the RNA primer become DNA. Dia akan tukar kepada DNA 1. So DNA primer will remove the primer from 5N1, replacing it with DNA nucleotide by added 1 to 1 3' end of the after last addition of the backbone. Okay, dia akan bind. Then after that, we will have the DNA ligase to rejoin the fragment. Kan? Karena kalau awak tukar ni, kan dia ada satu lubang dekat sini. So, we need to use the DNA ligase. So, got another enzyme is DNA ligase. So, DNA ligase ni dia akan join lah, rejoin again the DNA strand at here. Okay, so this is all about the DNA replication. So there are many enzymes you need to read. So hopefully you guys can know what are the function of the each enzyme. So this one is more or to the overall. So firstly, so I already mentioned. So I just recap again lah. So you guys can make a recap again. So this is the DNA, right? And then got one enzyme we call as DNA. Okay, got one enzyme we call as DNA helicase. Here got one enzyme we call as DNA helicase. So this enzyme will unwind the double-stranded structure of DNA. And then at the same time, we also have another enzyme called the single-stranded protein. So single-stranded protein is at here lah. Single-stranded protein. Betul kan? Single-stranded protein. Single-strand binding protein. Okay. Single-strand binding protein. So this one is just to stabilize lah. You want to stabilize the parental unwind structure one. And another enzyme is at here. At here. This one is DNA topoisomerase. So this one is just to main function is to form the circular DNA from the supercoiled DNA. Okay, so this is just the first step. I can say this is just the first step only. And then the next step is the formation of the leading strand. So want to form the leading strand, we need to find the origin first. So normally the origin replication is start from here, maybe. It start from here. So firstly, got one enzyme called RNA primase. So RNA primase will bind at here and then to form the RNA primer. So this one is RNA primer. Okay, after form RNA primer, got another enzyme we call as DNA polymerase 3. So got another enzyme use green. Okay, got another enzyme called DNA polymerase 3. Okay, so this enzyme will bind to the RNA primer. Not here. Bind to the RNA primer, and then it will form the leading strand. So it will form the leading strand through this direction. From this direction. So 5 to 3 direction. So you need to mention all of them is from through the 5 to 3 direction. So this is the function of DNA polymerase 3. Okay, next is after that, you need to form lagging strand. So how to form lagging strand? So normally lagging strand is formed from against the replication fork. So I use this color. So normally lagging strand is formed at here. Dia akan form at here, start at here. So this one is start at here lah. So for this one, it will start at here, opposite side. Okay, so same as the first step, it will form the RNA primase. So this gap they need to rejoin again. So how to rejoin? DNA ligase will play the responsibility to rejoin this band, this gap. At nucleotide lah, normally is nucleotide or form bond. Form bond rejoin. Okay, and finally it will form the new DNA lah. Yang ni dia akan form new DNA here lah. So this one is all about the mechanism of DNA replication. So yang paling penting is DNA replication enzyme, it involve many enzymes. So there are total 1, 2, 3, 4, 5, 6, 7 enzymes as well. So you need to memorize all these 7 enzymes. What are the function of the enzyme as well? So I think this enzyme is very easy for me. Even though I already graduated 2 years, I also can memorize. So I think you guys can memorize as well. Okay, so that's all for about the DNA replication mechanism. So hopefully you guys can learn about it. I know that this is very difficult. I hope that my explanation can make you easier understanding lah. Okay, so that's all for class today, I think. Okay, so that's all for class today. So any question or not? So no question at all. Okay, so everyone are expert. I don't know your school teacher already teach this chapter or not. So if you teach, so you maybe have some memory lah. So maybe more clear. If you not teach yet, so this is the first time. So hopefully you can understand. If not understand, you can ask your school teacher. Maybe your school teacher explanation more explain one more time. You can more clear. Okay, so that's all for today class DNA replication. So any question? No question. So okay, so just now I'm saying sorry that lost connection. So hopefully I hope that next time no lost connection at all. Okay, so that's all for class today. So thank you. See you next Monday. For those who retake sem 1 want to learn sem 1. If not, I see you next Friday. So thank you for attending the class today.